Hi, it's Dr. LeBlanc. I'm going to explain one of the absolutely key experiments in all of molecular genetics, Meselson and Stahl's demonstration that DNA is replicated semi-conservatively. DNA replication relies on the complementarity of DNA, the ability of A nucleotides to hydrogen bond to T and G to hydrogen bond to C, also known as Chargaff's rule. The process is something that's quite familiar to many students of molecular genetics. We know that the two strands come apart, that each serves as a template, and that the two new strands that are made are called daughter strands, and the two original strands are called parental strands. Back in Meselson and Stahl's time, there were three proposed models of DNA replication. The first, known as Semiconservative Replication, was a model that stated that the original DNA molecule, here indicated in purple, produced a brand new double-stranded DNA molecule where both of the strands of DNA were composed entirely of new nucleotides put together. After the first round of replication, then you ended up with a population of cells, say they're bacteria cells, with half of the cells with the original DNA molecules intact and half of the cells in the population with brand new molecules of DNA. After the second round of replication, you would end up with only a quarter of the cells in the population with the old original parental strands, and the rest of the cells in the population would have brand new DNA molecules in them, okay? And they didn't mix together. The other model of semi-conservative replication is much more familiar. In this model, we start off with a population of DNA with the parental strands of DNA molecule hydrogen bonded to each other, and that when DNA is replicated, these two strands come apart and each one serves as the template to synthesize a brand new molecule of DNA. Here we have an old parental strand and a newly synthesized strand of DNA together in a single molecule. After the first round of DNA replication, both of these cells, or all of the cells in the population, have DNA that is comprised of an old parental strand of DNA and a new synthesized molecule of DNA. If you let this population divide one more time, then you see differences in the DNA molecules between the cells. About half of the cells have hybrid molecules, which consists of a strand of DNA from the parental population plus a newly synthesized strand of DNA. Okay. And the other half of the cells in the population have only newly synthesized strands of DNA in their DNA molecules, because the generation previous to this, each of these two molecules act as parental strand. If you add a new strand to the light blue one, you get two light blue strands together. If you add a new strand to the purple one, you duplicate what the condition is in this generation. One of the original parental strands from way back up here, plus a newly synthesized strand of DNA. That's semi-conservative DNA replication. That's the one that we know already at this point. We know that this is the model that won out. The third model that was proposed was dispersive. In this model, the two parental strands somehow broke apart into small molecules, and the newly synthesized strands of DNA interspersed with the old molecules, so that at the first generation you had DNA molecules that were made of half of the old DNA and half new DNA, but the pieces were all interspersed between each other. After the next generation, you got a similar situation, except that now instead of half and half old DNA and half new DNA, you only have a quarter old DNA and 3/4 new DNA, but all of these molecules look pretty much the same as far as old versus new content is concerned. So there's no difference between any of these molecules. That's the dispersive method. Okay, here's a picture of Matt Meselson. If you want to hear him describe how he and Frank Stahl executed these experiments, you can go to the website DNA from the Beginning. That's what dnaftb.org stands for. And he's quite candid about how he and Frank carried out these experiments. Turns out they did them 3 times. One of them didn't work, and Matt is accusing Frank in there kind of lightheartedly of mislabeling the test tube. So it's kind of an interesting window into a time when all of these paradigms of DNA and molecular genetics were being discovered. Okay, so here's an overview of Matt Meselson and Frank Stahl's experiment. The experimental preparation. They grew E. coli in N15 medium for many generations. So they had a population of bacteria, E. coli, that had N15 incorporated into all the molecules inside the cell. DNA, proteins, whatever. Anything that had nitrogen, the isotope of nitrogen that was present in those cells was N15. Okay, normally, nitrogen is not this heavy isotope of N15. Normally, it is N14. Okay, these are not radioactive isotopes. These are just heavy and light isotopes of nitrogen. Okay, so here's the population of bacteria that they started out with. Here's the chromosome. It has 2 purple strands indicating that all the DNA in this initial culture of bacteria was heavy nitrogen. Okay, so to begin the experiment then, they took this culture of bacteria that had been labeled with N15 and they added to it nutrients that were labeled with a normal N14 isotope of nitrogen. Okay, so they added a solution of N14, which meant that it was all of the nutrients that they added were the normal light isotope of N14. They let the cells grow for various lengths of time. Okay, intervals equivalent to 1 generation, 2 generations, 3 generations. And then they removed cells from this one large culture of bacteria at various intervals, and they lysed them. Okay, what does that mean? Lysing cells means that you break them open so that there are no longer cells. There are just molecules. There are molecules of DNA in the cell wall. Okay, you lyse the cells by the addition of an enzyme, lysozyme, and detergent. It disrupts the bacterial cell wall and the cell membrane respectively. So instead of cells in the test tube here, they had a cell lysate, which means it's kind of a soup of broken open cells. Okay, now the next thing they did was a type of experiment that we don't normally do too often anymore. Okay, they loaded a sample of lysate onto a cesium chloride gradient. Okay, what is a cesium chloride gradient? Well, a cesium chloride salt, when you put it in to an aqueous solution and spin it in a centrifuge, not a microcentrifuge, a really big centrifuge, when you spin it for days at a time at very, very high speeds, high g-forces, we're talking 30,000 RPMs, which is equivalent in many cases to over 100,000 times the force of gravity. Okay, and you do this for 48 to 72 hours. When you do that, cesium chloride forms a gradient in the test tube. Okay, at the bottom is a very dense solution of cesium chloride. At the top is a very light solution of cesium chloride. It's a gradient from light at the top to heavy at the bottom. If you mix an organic molecule or a bunch of organic molecules like DNA with the cesium chloride while it's spinning, those molecules are going to migrate together into one section of the cesium chloride gradient, which is equivalent to their buoyant density. Okay, so you centrifuge the gradient until the DNA molecules reach their equilibrium densities. Since you have some molecules of DNA labeled with a heavy isotope of nitrogen and some molecules of DNA labeled with a lighter isotope of nitrogen, those two different types of DNA molecules will separate from each other, and that's what you see here. The lighter molecules will be higher up in the tube, and the heavier molecules will be further down into the test tube. Okay? And you can distinguish molecules of DNA that have two heavy strands or an equal amount of N15 and N14, or molecules that are half N14 and half N15, or molecules that are only N14, and you can distinguish all those different types of molecules from each other. You can observe it under ultraviolet light because DNA absorbs UV light. These are Meselson and Stahl's actual results. Okay, and in interpreting data from any figure, you always have to look at the labels. So at the top of these different panels are labels, 0.3, 0.71. These indicate generation times. Okay, so after 0.3 generations of growing the bacteria in culture, what do you see? You see most of the DNA is heavy, like the parental DNA. And you're gradually starting to see some half heavy, half light DNA here. Okay? If you go to 0.7 generations, you see more of the half and half. And after one generation of bacteria culture, all of the DNA is now half heavy and half light. It's all one type of molecule, half heavy, half light. This result is consistent with two different models. It rules out the conservative model of DNA replication, but it's consistent with both the semi-conservative and the dispersive method of DNA replication. Okay, the semi-conservative would indicate that you have one molecule of new DNA and one molecule of old parental DNA. Okay, so you've got a molecule of DNA that's half heavy and half light. The dispersive model of DNA also, shouldn't close that up at the bottom. The dispersive model also would indicate that you have half heavy and half light, but these two molecules look quite different from each other. In order to prove that it is or to give you, to lend evidence that it's the semi-conservative model and not the dispersive model, you have to let the culture grow for an additional generation. Okay, so this is what happened after, this is the results from the first generation. We have to go one more generation. So somewhere in between the 1.9 and the 3 here, well, let's pick the 2. It's a little easier to see. It's a more intense band. Okay, what happens at the second generation, after 2 generations? After 2 generations, we still have some of these molecules here that are half heavy and half light, but we also see molecules that are only light. Only light. And the only way you could do that is that if you ended up with, say, the blue strand here acting as template, the new strand from this molecule also acting as template and synthesizing a new strand off of that template. So you have a DNA molecule that has 2 new strands of DNA. If it was the dispersive model, you would get still more DNA molecules that were a mix of blue, more blue, and red. And all of the molecules in the generation, this next generation, would be the same. There would be the same percentage of old and new DNA. So this is after 2 generations. This is after 1 generation. So instead of seeing distinct bands here of half heavy and half light, or all light, you would see a single band in the middle that was a little higher up than this original band. After 1 generation, DNA is half heavy. That's consistent with both the semiconservative and dispersive models. After 2 generations, the DNA is of 2 types, distinct types: light, all light, and half heavy, half light. That's all light, and this is half heavy, half light, consistent with only the semiconservative model. And that's a sketch from their notebook.